Good morning, students. Today I am going to start a new chapter, that's chapter 6 from your class 12 biology syllabus. The name of the chapter is Molecular Basis of Inheritance. Already we have completed chapter 5, and from that chapter we came to know so many things about modern genetics and the genetical basis of inheritance. And from this chapter we will get to know the molecular basis of inheritance. So let's start the today's session, first session of chapter 6. So the first thing is the DNA and RNA world. So the first slide contains the DNA and RNA world. Actually, gene is the hereditary unit and is responsible for inheritance. Already we came to know from this thing from the chapter 5. And it's also well known that gene is actually the functional unit of the heredity. Okay, but what is the importance of this DNA and RNA? So what is the basic importance of the DNA and RNA? Because it's well known fact that gene actually a part of DNA and is made up of the nucleotides. All of you know that the segment of DNA is called the gene. I am repeating again, what is gene? The segment of DNA is called the gene. Okay, so actually a gene can grow, can reproduce and can mutate also. Okay, so this gene actually the ultimate version, ultimate unit of the recombination. Okay, and the genetic information also conveyed transfer from DNA to RNA. So that's why this DNA, you have to know about the DNA. You should know about the DNA and the RNA world. Okay, here actually the genetic material is one kind of the substance which controls the inheritance of traits from one generation to the next. Okay, I am repeating again, what is genetic material? You can ask me that thing, that what is the basic definition of genetic material and why DNA and RNA is the genetic material. So actually, first of all, you must know the definition of genetic material. So genetic material actually that kind of the substance which controls the inheritance pattern, inheritance of the traits of each and every characteristics from one generation to the next generation, from the parents to offspring. Okay, so next thing is the criteria. So by which criteria this DNA can be one genetic material, this RNA can be one genetic material. The first criteria is it should be able to replicate itself. I am again discussing this thing, that it should be able to replicate itself. So what is the meaning of this? The duplication of its genetic material by faithful replication, and it's actually the precise distribution among the new cells by the cell division. So what is the meaning of the replicate? Normal term of the replication, it means the xerox. Okay, so it should be able to replicate itself, it should be able to xerox itself. Okay, next thing, next criteria is the it should be stable both chemically and structurally. So this DNA and RNA has one structurally stable chemical and physical structure. So they have one stable physical and chemical structure. Okay, next thing, it should provide the scope for mutation. All of you know that it should be actually able to undergo mutation, and such a change should be stable inherited. Okay, so this is required for the evolution. If changes is not there in our body, so we will be stagnant at a point. So we need variation, and variation comes from the mutation. Okay, so already we have discussed this mutation thing in our chapter 5. Okay, so this is very much useful, this is very much crucial for the evolution. Next thing is the it should be able to express itself in the form of the Mendelian characters. What is the meaning of this? This Mendelian characteristics means the it actually follows the Mendelian laws. It means the law of segregation, law of dominance, law of independent assortment. Okay, so by these four criteria, one thing can be genetic material. So this DNA and RNA has these all those characteristics. So that's why you can say this thing that DNA and RNA is genetic material. But how you will prove this thing that the DNA and RNA is genetic material? Because this is science. Okay, so you need to provide one proof against this theory. So next thing is the search for genetic material. So the discovery of the nuclein by the scientist Mischer, and principles of inheritance by the Mendel was almost at the same time in the year of 1869 and the 1866 respectively. And actually these two things coincided, but for claiming that DNA acts as a genetic material took long time. Okay, so earlier discoveries actually made by Mendel and Sutton and Morgan and others had narrowed the search for genetic material to the chromosome. So by the year of 1926, the quest to determine the mechanism for the genetic inheritance actually reached the molecular level, because these scientists, all those scientists, the Mendel, this Sutton, this Morgan, all those scientists discovered the genetic material as the genetic basis in the field of the genetical study. But we need to reach this molecular level also. Okay, and gradually the question what molecule acts as the genetic material got answered. Okay, next thing is the transforming principle. Okay, this is the first experiment by which actually we get to know that the DNA is the genetic material. This experiment actually was conducted by the scientist Griffith. This experiment is conducted in the year of 1928, and this Griffith was a British medical officer which encountered a phenomenon. This is nowadays called the bacterial transformation. Okay, so why this is called the bacterial transformation? Because Griffith conducted these experiments to show transforming principles in bacteria. Okay, so that's why this is called the bacterial transformation. He conducted this experiment on mice, okay, and the pneumonia bacteria Streptococcus pneumoniae. So this Streptococcus pneumoniae is the bacterial pathogen for the pneumonia disease. So these for his experiment, these two things actually the subject, this mice and the Streptococcus pneumoniae. These bacteria are found in two strains. The first one is the virulent, that the smooth strain, and second one is the non-virulent, that the rough strain. So now I am coming to this thing that what is the basic definition of the smooth strain and what is the basic definition of the rough strain. It means the S and it is the R. So what is the meaning of the S strain? That this actually whose cells produce a capsule as their outer covering of the polysaccharides, causing the colonies on agar to be smooth and rather shiny. Okay, and these kinds of the strain is the pathogenic. It means, what does it mean the pathogenic? That these kinds of the strain, this virulent strain can infect the others organism, okay, and causes the pneumonia. Okay, so this is the smooth strain, S strain, and last one is the non-virulent strain, means the rough strain. What is the meaning of the rough strain? That in this case the cells lack capsule, okay, and produce the dull rough colonies and cannot infect the other bodies. Actually they do not produce the pneumonia. So this is called the rough strain or R strain. Okay, so I have written the definition also. The A strain bacteria produce capsule and is pathogenic, and R strain lacks capsule and is non-pathogenic. Okay, what is the meaning of pathogenic? Pathogenic means they can infect. They can infect the other organism's body. They can transmit the disease to the other organism's body. But what is the meaning of the non-pathogenic? That they cannot transmit the disease to the other organism's body. Next thing is this is his experiment, that first 1, 2, 3 and 4. Total 4 case study is there. Okay. So when we inject the rub strain, so that time mouse lives. Okay. Why? Because this is the R strain. This is the R strain, means the rub strain, and this is the non-pathogenic. Second one is when we inject the virulent strain, it means the smooth strain, that time mouse dies. The third case is when we are actually injecting the heat killed smooth strain, that time mouse lives. But when we are injecting heat killed rub strain mixed with the S strain, that time mouse dies. So this is the actually 4 case study. I have written also that when the R strains are injected into the mouse, it's a non-pathogenic and does not cause the pneumonia. The mouse continued to live. Okay. Next thing is when we are injecting S strains are injected into the mouse, that causes the pneumonia and mouse dies. This is the case study 2. In the case study 3, when the heat killed S strains are injected, that time they did not suffer from the pneumonia and thus they survived. And the last case study is when a mixture of the non-virulent and heat killed strain become mixed and injected into the mouse, that time it causes pneumonia and mouse dies. Okay. So this is the total 4 case study. Next thing is what is the conclusion of the experiment? What is the conclusion of the experiment? Because already observation part is done. But what is the conclusion of experiment? That R strain bacteria had been transformed by the heat killed S strain bacteria. Okay. The next conclusion is the transformation of the R strain to S strain is due to transfer of the genetic material. And the last conclusion is the biochemical nature of the genetic material was not defined from his experiment. Okay. So we need to know the biochemical nature of the genetic material. We need to know the biochemical nature of the genetic material. So the Griffith actually effect gradually become the known as the transformation and turned out to be the first step in the identification of the genetic material. But the chemical nature of the transforming particles not proved. Actually, he discovered the transformation procedure of the genetic material, but he could not identify the biochemical nature of the genetic material. Okay. So from his experiment, this is the failure of his experiment. Next experiment is the biochemical characterization of transforming principle. I am repeating again. Next experiment is the biochemical characterization of transforming principle, because already Griffith experimented this transforming material. Okay. But now we need to know the biochemical characters of the transforming principle. In the year of 1944, 16 years after Griffith's experiment, actually, there were 3 scientists, Avery, MacLeod and the McCarthy. Okay. So, in the year of 1933 to 1944, they worked to determine the biochemical nature of the transforming principle of the Griffith experiment. They took 11 years for their experiment and they tested actually the fractions of the heat killed cell for transforming ability, and their finding actually, so they purified the biochemicals from the heat killed S protein and they got the protein, DNA and the RNA. Okay, and after that they added some digestive enzymes of each to see which one could transform the live R cells to S cell. Their findings actually were DNA alone from the S bacteria caused the R bacteria to become transferred. So there is total 3 case studies there. Okay, so heat killed S strain plus protease plus live R strain, then it transform R strain to S strain. Okay. Next case study is the heat-killed strain plus RNase enzyme, then live art strain. It also transforms art strain to A strain. And the last one is the heat-killed A strain plus DNase. There is another enzyme is there, DNase plus live art strain. Then actually it unable to transform art strain to A strain. Okay, so this is the, so for that DNase enzyme, the art strain cannot be able to transform to A strain. Okay. So what is the conclusion of the experiment? The first thing is the protein of heat-killed A strain is not the genetic material. I am repeating again, protein of heat-killed A strain actually is not the genetic material. The second one is the RNA of heat-killed A strain is not also the genetic material, and DNA of heat-killed A strain is the genetic material. Okay, so DNA is the genetic material, here protein. So when we are using protease, it means the protein. So it's not the genetic material. Why? Because it's actually transform R strain to S strain. So when we are using the RNA of the heat-killed A strain is not the genetic material. Why? Because when we are using the RNase enzyme is coming from the RNA. So when we are using RNase enzyme, so that time also R strain transform to the S strain. So it means it's not the genetic material. But when we are actually use the DNase, that time the RNA strains cannot be able to transform to S strain. So that's why you can say this thing, that this is the genetic material. This is the genetic material. So because DNA digested with the DNase mixed with the R strain, unable to transform R strain to S strain. But all biologists are not convinced. Okay, so they finally concluded that DNA is the hereditary material. Why? Because the digestion with the DNase enzyme actually inhibited transformation procedure. So they finally concluded that DNA is the hereditary material. But all biologists are not convinced. Next is the genetic material is DNA. There is another experiment is there. Okay, so all of you know that the viral infecting agent is DNA. Okay, after that, by using the radioactive tracers, Alfred Hershey and the Maratha Chase, Hershey and Chase in the year of 1952 gave evidence that the DNA is the hereditary material in certain bacteriophages. Okay, bacteriophages is the bacterial viruses. They worked on the virus actually that infects the bacteria called the bacteriophage. I am repeating again, they worked on the virus that actually infects the bacteria called the bacteriophage. Okay, just look at the diagram. This is the bacteriophage, and I think all of you know this bacteriophage because already you have studied in your class 11 standard this bacteriophage, about the bacteriophage. So during the infection, the bacteriophage actually attaches the bacterial cell wall and it inserts its genetic material into the bacterial wall. Okay, so this is their replication and reproduction procedure. This is their replication and the reproduction procedure. So after that, actually Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. So actually they conducted their blenders experiment on the T2 bacteriophage which attacks the bacterium Escherichia coli, and the phage particles were prepared by using two types of radioisotope. So they grew some viruses on the medium having the radioactive phosphorus and some others on medium having radioactive sulfur. Okay, so they are using two radioactive isotopes. One is the radioactive phosphorus and another one is the radioactive sulfur. So viruses grow in radioactive phosphorus, have radioactive DNA but not radioactive protein. Because phosphorus present in DNA, not in the protein. After that, the viruses grows in the radioactive sulfur have the radioactive protein but not in the radioactive DNA, because sulfur present in the protein but not in the DNA. So these phosphorus are present in DNA but sulfur is absent in DNA, because phosphorus, this DNA has the phosphorus backbone. Okay, so this is the actually the picture. The protein coats of phages are radioactively labeled. After that, phages infect the bacteria with the genetic material. After that, the no radioactivity enters the cell. Okay, after that, this is case study 1. Okay, and this is the case study 2. Here the DNA of bacteriophage radioactively labeled. Okay, second one is the phages infect bacteria with the genetic material and radioactively entered cell. Radioactivity enters the cell. So this is the 2 case study. After that, the viral, after the radioactive phages were allowed to infect this E. coli bacteria, that time the phages transferred the genetic material to the bacteria, and the viral coats were separated from the bacteria surface by the blender's machine. These virus particles were separated from the bacteria by centrifuge machine actually. Okay, so what is the observation part? That the bacteria infected with the viruses that had radioactive DNA were radioactive, no radioactivity in the supernatant. Okay, and the second observation is the bacteria infected with the viruses that had the radioactive protein were not radioactive, but radioactivity found in the supernatant. Okay, so what is the conclusion? What is the conclusion of the experiment? That DNA is the main infecting agent. So towards the end of the eclipse period, fast DNA actually directs the production of the protein coats assembly of newly formed fast bacteria. Okay, so what is their conclusion? The main conclusion is that DNA is the infecting agent that made the bacteria radioactive. Hence DNA is the genetic material, not the protein. Okay, so because in the biochemical nature actually so many biochemical product is there, like nucleic acid, like protein, like another amino acid is there. So you have to prove this thing. Actually the scientists had to prove this thing, that DNA is not the protein, this is one kind of the genetic material. Okay, and this DNA is made up of by the nucleic acid. So this is the conclusion of the experiment. This is the conclusion of the experiment. So this is your today's class. From this class actually we came to know that how all the scientists discovered that DNA is the only main genetic material present in our body. Okay, so in our next class we will discuss the properties of the genetic material, the main structure of the genetic material. Okay, so because all of you know that they have the some properties which actually clarify this thing, that DNA is the genetic material. Okay, so in our next class we will discuss the structure, the properties of the genetic material, it means the nucleic acid and the DNA. Okay, so from this chapter, actually this chapter is quite complicated chapter but quite interesting chapter. So please go through each and every session of this chapter, and in the meantime if you have any queries, so ask me immediately. Okay, thank you, bye.